everybody. My name is Diego Hernandez, and it is my pleasure to be with you today. Thank you so much for the introduction, Dr. Jander. And before we begin, I want to introduce somebody who I think I have a little bit of a love-hate relationship with. That is my friend over here on the screen. His name is MM01545. And I did some phenotyping work over the spring at the Frank Lab, and he got me a lot. I think that's why I hate him, but he looks super cool, so I have some love for him. And I think he was the inspiration for my project. And essentially, he pricked me, and it caused me to ask myself, what are the genetic loci involved with the evolution of eggplant prickles? So now, before I get into the project, let's do a little bit of background and general information on eggplants. Now, they are within the Solanaceae, or the nightshade family, and they're actually the third most consumed behind tomatoes and peppers. And the center of origin is thought to be around Southeast Asia, so maybe Thailand, Malaysia kind of area. And this is a contested piece of data. It's still being developed based on genomic data. And then today we're going to look at 3 specific species within the eggplant family. And that's going to be Insanum, Ovigerum, and Melangina. And as the slide says, this is kind of a good breadth of some close wild relatives, an intermediate, and then your more cultivated eggplant. That being said, we're going to look at the first player in the game. This is Insanum. He's kind of like that jock in high school, you know, big, mean guy. A really bunch of prickles, smaller leaves, smaller flowers. The fruit were mean looking, small as well. And there was a high seed density in the fruits and not so much of a fleshy interior. Ovigerum, I think, was a little bit nicer. Ovigerum might let you like sit next to it in lunch hall, but overall still had some more of the wild tendencies, I think. Maybe a little bit larger leaf size, a little bit larger flower size, as well as fruit. And you can see there is some more flesh, and they're also seeing an increased carpal number a little bit in Ovigerum as well. The final, and one of my favorites, is Melangina. Melangina is more of your typical eggplant. As you can see, there's a general reduction in prickles, as we'll see today, larger flower and fruit size. And I think the fruit diversity in Melangina is just remarkable. And I'll use this time to give a little shout out to my colleague, Edward Almanzas, who did a phenomenal study on fruit eggplant morphologies. And then, of course, you're going to see lower seed density, as well as larger fleshy material in the fruit and increased carpal size. Now, with that being said, I want to give a brief segue to an overall eggplant genome project, which is being headed by a postdoctoral researcher, Hamid Razafard. And this is within the Frank Lab. And we are sequencing about 100 extensions from around the world to kind of get a breadth of the variety of eggplants. And these seeds are coming from INRA, which is a seed bank in France, as well as the USDA. The acronyms there, MM and PI, are going to determine which seeds came from which seed bank. And the sequencing behind this project, essentially, it was a lot of short read sequencing, so Illumina of the majority of the eggplants, but then it was packed by a long read sequencing on a special eggplant, Eggplant 6, which is actually transgenic, which they utilized to set up a new reference genome for eggplant. So it's really exciting. There's also a gene expression analysis aspect for this project. And again, we're just looking at the transcriptomics behind eggplant varieties. And then there's the phenotyping side, which is where I came in. And we did a lot of phenotyping over the spring and then as well in the summer. During the summer, we grew our plants on the Thompson Research Farm. And these plants are still maturing. We're hoping to get some more data from them later on. Now, I want to give just a brief geographic distribution as to the home for these seeds. So these seeds all came from the seed bank, but the seed bank gave us information as far as where these seeds were collected from. And we're going to see that we got a lot of the seeds from the proposed center of origin. So around here, maybe Southeast Asia. And the mentality behind that was so we could capture as much of the variation around the center of origin. That's where we believe are going to be the most diverse species around there in eggplant. But of course, we got some eggplant from countries surrounding as well, as far as Russia and Africa as well. Now, when we think about prickles as an agronomic trait, there's an interesting story to be told. On one side, there's a general push for a loss of prickles, mainly because, number one, they hurt. And then as well as there can be some sort of mechanical harm in post-harvest processing of fruits from the prickles of another fruit. But then there are some regions in India which actually have a preference for prickles on fruit calyxes, which is this top part of the fruit here. And it's thought to believe that there's a consumer perception behind prickle presence on the calyxes. Seems that they prefer, it may have some more organoleptic qualities, which is just your fruit, color, taste, texture kind of thing. And it's perceived to be higher on eggplants that have some prickles on it. But overall, the general trend is a reduction in prickles. And I wanted to briefly highlight some studies around prickle development in eggplant. And they found some QTL regions on chromosomes 2, 3, 6, and 7 that were of interest, and a transcriptomic study in developing prickles found some potential candidate genes. The first having a homolog in Arabidopsis for trichome development, and the second, something similar as well, was an auxin response factor. And these are just potential candidates. Hopefully, part of this project is to see if we could find some more. Now, before we get into the project itself, I wanted to show you where I began. And I began with understanding the complexity behind the phenotype of prickles. And so here we have one of the more wild, one of my good friends, 1545. And as you can see, it's really gnarly looking, really mean, bunch of prickles all along the midrib here, and as well as veins and on the stem. And you just see it's a bunch of prickles and they're very large. On the other side of the spectrum, we have the cultivated eggplants, which generally do not have prickles, none on the midrib, none on the veins, and none on the stem. But then, of course, life is never black and white. It became a little bit more complex. We had some semi-cultivated, which were somewhere in the middle of that spectrum. You can see here there are prickles, but they don't seem to be so much on the veins. It's mainly just the midrib, and the prickle size is reduced, especially in comparison to the leaf size. And there's a lot less prickles on the stems. So this is where the story of prickles started to develop in my head. And I'm thinking, okay, how are we going to look at this? And that's where the outline of my project began. Now, of course, like I said, I wanted to understand prickles. I wanted to become the prickle. So we began by doing a developmental imaging aspect to the project. And we looked at some of the extreme phenotypes, so very prickly and not so prickly. We had hoped to do some X-ray CT scans, but unfortunately the data didn't come quite in time. It actually came in just today. So to substitute that data, we did some dissecting microscope work, which will hopefully give us a good idea about that. But essentially, after imaging these young eggplants, we were able to understand the phenotype and then begin developing a scoring method. And so we scored the leaves and flowers for these eggplants. Following that, we're going to use maybe that phenotypic data in conjunction with some genomic data and see if we can begin to tell the story of eggplant prickle evolution. So now I want to see, the imaging aspect. How can I use imaging to understand the prickle phenotype? So like I said, we did the extreme. So we had a non-prickly eggplant and an extremely prickly eggplant. We planted them, had them germinate, and then right around the 4th differentiated leaf stage. So this is after the cotyledons, 4 leaves coming out. We took it out, and then I put it under the microscope and tried to take some images. And so we're comparing here the first differentiated leaf. So this is the first leaf the eggplant puts out after germination, after the cotyledons. And on the left, we're going to have the non-prickly variety. On the right, we have the prickly variety. So as you can see, this is the first leaf. This is very important for the plant. And it seems like in general, there's not a lot of prickliness going on in these leaves. And a hypothesis might be because the plant is prioritizing other things, such as maybe photosynthesis and whatnot. But then things got a little bit interesting as we looked at the 3rd and 4th differentiated leaves. So on the left side, on the non-prickly variety, this is the 3rd differentiated leaf here, and this is the 3rd differentiated leaf on the prickly side. And what you can see is this leaf is younger essentially than the 1st differentiated leaf, and we are seeing large prickles. Here, there are even 2 clumped together, as well as trichome development and other things and whatnot. But it begins to tell the question: at what point does the eggplant start prioritizing these prickles? And then the 4th differentiated leaf, on the non-prickly, it's here. I was able to get them both in the same image, and it just looks like a little fuzzy guy in the corner. And then in the prickly variety, we saw something very interesting. So this is essentially a very young leaf. It has not even expanded yet. And if you look closely, you can see there's a pretty substantial prickle already forming in this young leaf before it's fully expanded. So essentially, it's doing even little photosynthesis at that point. But the plant is now very protective over its leaves, and so it's releasing those prickles. And these were just interesting things to look at, interesting things to keep in mind. But overall, it allowed me to understand and, like I said, become the prickle. And with that being said, we started now doing some scoring. Our scoring became very simple at first. It was pretty much just presence or absence. So we looked at the leaves and flowers of these eggplants, and we looked at scanned images. We cut off the 3 full first fully formed leaves from each section, and then saw if it had prickles or not. And then we looked at the number of leaves that we had cut, and we said, do all 3 leaves have prickles or only 2 or only 1? And we gave that a score. Then we looked at the flowers as well, and we did something similar. We cut off 3 of the flowers that we felt were at a similar developmental stage, and then we counted to see if the 3 flowers had prickles. So for example, if you were doing research with me this summer, you would have looked at a lot of these images and counted 1, 2, 3 out of 3 flowers. Well, this one only has 1 here, some small prickles there, and then appears to be prickleless on the other 2. So now you guys can go and phenotype anything. And what we got from that data was kind of general. We had 89 eggplants phenotyped overall, 61 had prickles and 28 did not have prickles. Of the ones that did have prickles, about 43 or so had some on the flower, 18 had flowers and leaves. But interestingly enough, there were no eggplants that had only prickles on the leaves. How is that going to play into the data? We'll see, but that was just one of our interesting observations. This data does a little bit more for us when we put it through a filter of species. So on the bottom axis here, you're going to see the 3 different species of eggplants that we looked at. And then on the left axis, you see the proportion of the species that had the specific phenotype. And as you can see here, Insanum obviously was going to be more wild and have much of its plants be prickly, both in the leaves and flower. Only a small portion of it didn't have any prickles at all. Ovigerum, which was thought to be somewhat of the middle semi-domesticated stage, interestingly enough, was essentially less prickly than Melangina. And what that means is going to be almost a story to tell, but if I had to hypothesize, it may seem like there's some selection going on in the Melangina population that is leading to retaining prickles on the flowers, but also removing prickles on the leaves. But we'll do some further analysis on that as we continue forward. And I wasn't satisfied with just the presence and absence. Felt like I needed a more comprehensive view as far as maybe more of a prickliness factor. So I came up with the scoring for prickle density. And essentially that was the number of the prickles on the top and bottom of the leaf added together and divided by the leaf area. We were able to accomplish this in ImageJ, which is an image analyzing software. And then we started to get some more interesting results. This is just a flat out result for prickle density by species. Of course, as you can see, Insanum is going to have a much higher prickle density, so more prickles and smaller leaves. Ovigerum, interestingly enough, only had 1 plant that had prickles on the leaves. This could be that an early ancestor for Ovigerum was just reduced prickles, and then as it radiated out, it continued that phenotype. But it does give an interesting story to tell. Melangina, of course, had a decrease in comparison to Insanum. And then once we had all that phenotypic data, we decided, okay, we also need to take some genomic data. And this genomic data came from the sequencing of the 100 accessions from around the world. And once we did the sequencing on that data, we needed to do something called variant calling, which allowed us to essentially compile the differences in genome between all of these eggplants. And the differences were going to be in terms of SNPs, single nucleotide polymorphism, as well as insertions and deletions. Once we had that, we needed to adjust the data a little bit because essentially if there was an allele or a variant that was found in less than 2% of the population, so in 2 plants in the homozygous state or 4 plants in the heterozygous state, that could have easily just been due to low DNA quality. So we went and we removed those from the data set. And then once we got a nice clean data set, we were able to use those to construct different genomic analysis. The first one we did was a PCA, a principal component analysis. And this is just a good illustration of the variation amongst all of our eggplants. Essentially what we have is 2 dimensions on this graph. And these dimensions were able to capture roughly about 30% of the variation among eggplants. And what we're looking for is the clustering. So as you can see, the Melangina, which are the purple dots, are clustered together. They're very similar in terms of genetically, in terms of their genetic structure. The Ovigerum are a little bit less clustered, but still close together. And then the Insanum are all over the place because it's to be presumed that the more wild varieties are going to have more untapped genome structure and much more variety when we look at things overall. So this is a good visualization tool. The next step from here is to start constructing phylogenies. So we were able to do that. And here you see a phylogeny of all the eggplants that we were able to sequence. The things that you want to look at in terms of this phylogeny is the grouping. So obviously here we have a very wild group here made of mostly Insanum and some Ovigerum. And then of course here we have a very extensive Melangina, or cultivated group, all grouped together. And this is going to begin to tell us a story about how these eggplants traveled and where they came from and whatnot. And the Melangina that are almost sparsely located on other parts of the phylogeny, that story might be because of different hybrid crossing that might have happened early on in domestication for that plant, or it could be something like a regional exposure, geographic exposure. And so when we take this data and we add some of the phenotypic data to it, things get a little bit interesting. So what you're seeing here is red dots are eggplants that have prickles on the leaves and flowers, and then pink dots are eggplants that only have prickles on the flowers. And as it is expected, the wild ones are more likely to have prickles on the flowers and leaves, and then it seems as though the Melangina are more likely than not to have prickles, but only have prickles on the flowers. Overall, though, we want to look at the trend that this phylogeny shows us, and we're going to see an overall reduction in prickle phenotype, ultimately resulting in just prickles being on the flowers. The next step in the project was, okay, now we need to start looking and seeing where exactly might the loci be for prickle phenotypes. And essentially we were able to accomplish that by doing a genome-wide association study. And a GWAS works very simply. You're going to take an input and it's going to give you an output. The input is very important. So our input was our variant data, so our SNPs, our insertions and deletions. And then we compiled that also with our phenotypic data, so our presence and absence, and then our prickle density. And then what it's going to output a correlation analysis between the variations and the phenotype. And that's going to help us to understand where exactly in these genomes are the genes associated with our phenotype of interest, in this case, prickles. So we had presence or absence, and essentially what you're seeing here is called a Manhattan plot. We have the genome of the eggplants, 12 chromosomes long, on this bottom axis. And then on the left axis, we have it's a p-value. And essentially the p-value has to be of significance. And since we're essentially running a correlation analysis on 7 million SNPs and insertions and deletions, you might get some random hits. So by setting up a higher threshold line, we're able to take out some more of the noise there and see exactly what's going on. So as we did the presence and absence GWAS, we saw a region of interest on chromosome 6. And that's denoted by this almost like long hike here and then some significant SNPs there. And this long hike is due to linkage disequilibrium. So this is just trait of interest dragging on some regionally close genes with it as well. And so this was interesting because other papers I had read had also seen something on chromosome 6. I kind of had this aha moment, like, oh wait, this whole science stuff actually works. You're validating other people's data and whatnot. It was a little bit exciting. When we did the prickle density, we did find something new though. We found a region of interest on chromosome 11. We're not sure what this is yet. That'll be some future things to look at. But again, it is exciting. This could be a potential different loci associated with prickles. Overall, though, in terms of the conclusions for this project, there was, of course, the gradual loss in prickles in terms of eggplant domestication. But of course, the S. ovigerum story is a little bit interesting. And I wanted to put the map here to show you that a lot of the ovigerum are found here around Thailand. And it could very well be the case that the initial ancestor for this group was maybe reduced in prickles or didn't have prickles. And then as that group grew out, that trait maintained, but of course it has many of the other wilder traits for eggplants. Of course, we had some other novel ways to develop phenotype prickles, such as the leaf density. One way that I think would be good to do in the future is maybe the prickle length. So essentially snipping off the prickles and measuring those and then running a GWAS on that, seeing where we get hits in the genome. And then of course, we had the regions of interest on chromosome 6 and 11, which are gonna be potential targets for future breeding, maybe transformations as well, and hopefully we can further illuminate the genetic story around these loci. That being said, I would like to thank the Frank Lab, and specifically, this is we call ourselves Team Melangina. From left to right, we have Run Long, Paul, Ashley, Sierra, Eddie, Margaret, Frank, Elise, myself, and then Hamid, my mentor. And then of course, the Frank Lab, see them sitting over there in the back. And I would love to thank Delaney, Olivia, BTI for this wonderful opportunity.